So we'll have the practical lab on Saturday, I guess. Saturday is a Monday, right?
Yes,
Yes,
ma'am.
ma'am.
Hmm. Okay, so today's class is on the introduction to the bacterial strains, how they are engineered, and subsequent to that class, we'll start with the cloning vectors, which mainly fall in two categories, plasmids and fudges. So now let's say we are delving into recombinant DNA engineering. Okay, so with respect to bacterial strains and the whole this cloning strategy, let me just apprise you that what is the how do you clone your gene of interest to get your recombinant protein, and then I will tell you that about the bacterial strains. Anyway, so we had discussed that the various enzymes which are used in molecular biology, which you will find are helpful when you have to actually perform cloning. And first thing is you have to get your gene of interest. For that, you have to perform PCR. Generally, you have to perform PCR, keeping the primers at the ends of the DNA that you want to amplify. They should have a restriction enzyme site, and the details for that I think are in one of the assignments that has been shared with you. Then you have to ensure that the restriction enzyme sites, it's preferable that on one side you have one restriction enzyme site and on the other side you have some other restriction enzyme sites, just to ensure that when your DNA gets ligated into a vector, then it is a directional cloning. However, even if it's not a directional cloning, there are ways to look for the recombinant plasmids in which your gene of interest has gone in the right direction. But preferably, you should have two different enzymes on either side of your gene of interest. So one way is to perform PCR, and I'll revisit PCR in one of the lectures. But another way is also that maybe your gene is already cloned or present in some DNA, some recombinant clone, and you just have to cut it enzymes and then just subclone it in another vector. So two ways of getting foreign DNA is just by doing restriction enzyme digestion or by PCR followed by restriction enzyme digestion. Then you have to find some way of transferring your gene of interest into bacteria. And is it only bacteria? No, you have, see, your ultimate aim is to get more copies of this DNA, or your ultimate goal could also be in majority of the cases to get this gene of interest amplified and giving you recombinant protein in huge quantities. So that is why we want that it should go into some organism which will be able to replicate it and further transcribe and translate it. So one of the most common used host strain is bacteria. Why bacteria is most commonly used, I just come to that. But then the idea is that if you incubate DNA with bacteria, then you have to find a way by which this DNA it is very efficiently gets into bacteria, and not only gets into bacteria but also makes more of its copies. So just a fragment of DNA going into bacteria will not help. You will actually require a vector. So what that vector could be, it could be plasmid DNA, it could be phage DNA. Why plasmid DNA and phage DNA? Because they both have the capacity to make more of their copies once they are inside bacteria. Just getting this fragment of DNA into bacteria will not help. I'll just tell you this point again, because you want more copies of it to be produced. Why plasmid can make more of its copies? Because it has its own origin of replication, and it requires certain proteins from the bacterial host strain to replicate into multiple copies. Once you have multiple copies of this plasmid or recombinant plasmid, then obviously you can harvest either the DNA in more amount, or if your ultimate aim is to harvest the recombinant protein, then you can harvest the recombinant protein also. Now you have to, once you decide which vector you want to use, it could be plasmid, it could be bacteriophage. Why bacteriophage? Because even bacteriophage has the capacity to inject its DNA into the bacteria, and that DNA can make more of its copy. And I've already discussed that regarding lytic life cycle. So you have to find a way to introduce your foreign DNA into plasmid DNA, and with the molecular biology tools which I've told you in past 2-3 classes, what you can do is you can cut your plasmid with a restriction enzyme, you can cut your foreign DNA with a restriction enzyme, and then you ligate these two together with the help of a DNA ligase. Which different ligation strategies you can use in a particular scenario, I've already discussed that in much detail. So once you have a recombinant plasmid made, then you have to find a way to introduce into bacteria, and those ways, the most common way is transformation, because in case of at least E. coli bacteria, transformation works very, very fine and it's very well standardized. You can also use electroporation, you can use multiple strategies for introduction of your DNA into the host organism, depending on which host organism you are using. It could be mammalian cell, it could be plant cell, it could be bacteria, and so on. And which different techniques have been standardized. It could be electroporation, it could be bombardment, it could be, I've told you, transformation, etc. So once this thing is inside the bacteria, it will make more of its copies. Then you have to find a way that which bacteria has taken up the plasmid. So you have certain selectable markers, and then you have certain screenable markers. And I'll come back to these 2 points in much detail later. And then only you will be able to identify the bacteria which has got the recombinant plasmid. Once you have the bacteria which has the recombinant plasmid, it is like your cell culture factory, and it will make lots of your protein of interest, which you can harvest and further purify for either your own molecular biology purposes in the lab, or you can be a owner of a company and sell it as an enzyme to be used in molecular biology for basic understanding or for manipulation purposes, or you can sell it for therapeutic purposes, various therapeutic purposes where proteins are used for administering proteins, or enzymes are used for treatment of various diseases. So yes, so this is the general scheme which is used for recombinant DNA technology, involved in recombinant DNA technology. Now coming to that, let's say you are owner of a company and your ultimate aim is to make lots of bacteria, lots of recombinant DNA, this protein, to be sold to various scenarios. So your idea would be to use, like, minimum input should be there, means you that not much money is used. It is much cheaper to produce the protein, and means lots of protein should be produced. These are the two major points. So and even time is a constraint, right? So with all these points in mind, bacteria comes as a very good host strain, and in many cases bacteria is used as a host strain, be it in your lab for basic understanding or be it in the various industries. So why bacteria is preferred as a host strain, and later I'll come to why it is not preferred as a host strain in certain scenarios. But let's come to why it is preferred as a host strain. One, that it is very easily grown. It just needs few chemicals to be grown in the lab, and you know that how cheap that uh, LB is, and bacteria just grow very fine in that. And you will realize that what is the difference between maintaining bacteria and growing it in a lab versus if you are using a mammalian cell. So mammalian cell maintenance requires lots of money, much, maybe 100 times more than just maintaining bacteria. So that way it is like very easily grown, and it is very well standardized that under which condition we can have optimal growth of certain bacteria. And I've already told you they are cheap to grow. They grow very fast. So I've told you with from industry point of view, even if I forget your basic experiments in the lab, from industry point of view, their fast growth is very, very important. Even like maybe 2 3 hours makes a lot of this difference when we are talking in terms of industry. Then they are easily manipulated. You might have seen that in this case, we have to find a strategy in which bacteria takes up the foreign DNA. So in case of E. coli and other bacteria, which are these techniques are very, very well standardized. I'm sure that in your molecular biology practicals, you've already done transformation and you can understand how easy it is to perform transformation. So these things also become very important. You don't want a host strain in which you are struggling. It means you have made your recombinant DNA, but you are struggling to introduce into a particular host strain. So this is a very important point. Then once you have made the recombinant DNA, the DNA should also be easily isolated. So I'm sure that in molecular biology practical, you've also done how to isolate DNA, be it plasmid DNA or bacterial DNA from bacteria. Then there are generally non-pathogenic in nature, or even if they were pathogenic in nature and were serving as a very good host strain, they have been manipulated in such a way that they are non-pathogenic. Then one very, very important property that is involved is that whatever you are cloning into bacteria, your foreign DNA, it should be extremely stable in nature. It shouldn't be that you use, so much of resources and time in developing a recombinant strain, but finally you find that it is lost over time, or it is getting a lot of recombinations and mutations are taking place in that. So in that context, bacteria is a very good host strain, and generally whatever you clone within bacteria, they are found to be extremely stable once they are incorporated. So this is also a very important point. And at this point, maybe you should have come here. They contain natural plasmids and viruses, which can be used as vectors for recombinant DNA. And we'll revisit these 2 points at various scenarios where I'll be telling you about the various cloning vectors. Then also they are equipped with appropriate enzymes to allow replication of the vector, followed by transcription and translation. So with all these points, definitely bacteria come as the first choice for expression of any recombinant protein. And I guess you might be knowing that generally bacteria are not used as host strains for expression of eukaryotic proteins, and especially for those eukaryotic proteins which require post-translational modifications. And when I'm talking about post-translational modifications, I'm talking about the various different post-translational modifications, without which certain proteins, even if they are produced in huge quantity by whole this process, but unless these proteins they have post-translational modifications, they will not be functional. So if they are not functional, they are of no use. So with all these points in mind, yes, there are certain constraints with respect to E. coli, mainly it being a prokaryotic organism, and many a times you do not prefer it for expression of eukaryotic proteins, especially those which have post-translational modifications. Now coming to that, you find a bacteria strain which is very good in all these aspects. But will you be like happy with that strain? I've told you that it's a dynamic field and people want to, improve over the existing strains for various purposes. What those purposes can be, there can be many different purposes, keeping in mind that your ultimate goal is to get sufficient amount of recombinant protein. So many of these bacteria, they have been engineered in by various ways and for various genes, so that the final recombinant protein you get enough of it, and also it is highly stable in nature. So let's see one by one that which are the various modifications that have been done in these various bacterial host strains. It could be E. coli, it could be Bacillus, etc. And how they have helped in final recombinant protein production. So these are some general changes that have been made in these genes that are mentioned here. So let's see the OMPT. So strains which are harboring this mutation, they are deficient in outer membrane protease this 7, and why it has been engineered so that this protease does not uh, is not produced or is not functional is because we don't want the proteolysis of the expressed recombinant protein. So many of the engineered host strains that are commercially available in the market, you will find that they have mutation in this gene or in this gene for the similar purpose, and or in these genes which are mentioned here. Please go through these all points. So what you want is that when you are introducing your foreign DNA, it should not be restriction digested. I've told you about restriction methylation system in my previous class. And obviously, you would not want that any foreign DNA that you are introducing, either it should be methylated or it should be restriction digested. So for all these purposes, you prefer a host strain which is deficient in these systems. So be it for this system or for this DCM or DAM system, which is involved in methylation of certain residues. So these are some points. If you go to any commercial bacterial host strain website, you can just see how many negatives or positives they are written as saying that the host strain have been engineered in various ways to make it much better. Now let's consider, or let me tell you about the various other modifications as well. I've just told you that you don't prefer a host strain which is restriction or methylation positive, or it should be proteolysis deficient, etc. But there are other changes also which have been done in these bacterial strains. So one of the very, very commonly used bacterial strain is BL21. And in this, what they have done is, with the idea that you want to express a protein in an inducible system. So in an inducible system, it was realized that, okay, IPTG is a very good inducible system, but at the same time, they also want a very, very strong promoter. So what they have done is that they have introduced a T7 promoter upstream of the any gene of interest that you are cloning. Okay. Now, because you are introducing a T7 promoter in your plasmid, in your plasmid vector, so because it is E. coli, it is a bacteria, and T7 promoter belongs to a bacteriophage, then obviously T7 promoter means it will not be recognized by E. coli RNA polymerase. So E. coli RNA polymerase, if it is not able to recognize the T7 promoter, then obviously the transcription will not take place. So what they have done is they have engineered the bacterial strain in such a way that it is expressing the T7 RNA polymerase. So BL21 strain, not only it gives us an inducible system, but also a very strong promoter system in which RNA polymerase is very, very proactive, and whatever you have cloned is produced in huge quantity. So that's why it's a very preferred strain in, and you will find it in many labs which are performing recombinant DNA proteins. So it is a little complex how this whole system has been made, but I'll tell you in much more detail when I'll be telling you about the expression vectors. Okay. So, but for your information, I just want to tell you that for an inducible system and a very strong protein expression system as a final use, BL21 strain has been developed. But in many cases, there is a possibility that the gene that you are cloning may have some toxic properties. because of which if you express it in this strain, there is a possibility that it will lead to, you know, decrease in the final output because it was toxic to the bacterial strain. So in those scenario, what they do is that they try to limit the expression of toxic protein at all times, which is obviously done because it's an inducible system. But to make it even more clean, they have introduced a T7 lysozyme, which ensures that the expression of T7 RNA polymerase is comparatively limited as compared to this strain. And there is no basal expression of T7 RNA polymerase under non-inducible time. So because of this, only when you want to express the protein, only then the protein will be expressed, but not to that high levels which were produced here. So again, this strain is also very commonly used in many labs, especially where they find that the protein was little toxic to the bacterial strain. Now to introduce another, they have introduced T7 lysozyme here, which is expressed as part of a plasmid. So it means that in this bacterial strain, they have introduced a plasmid already, which is expressing T7 lysozyme. So now, because you are introducing a plasmid already, and now you want to introduce your plasmid, which so I would say that the bacterial host strain BL21, which is having that T7 lysozyme, it already has a plasmid. Now let's say you made a recombinant DNA plasmid in which your gene of interest is there and you want to introduce it into bacteria. What you have to ensure is that these two plasmids, means the plasmid in which you have introduced your foreign DNA and the plasmid which is already present in the bacteria, which is having that T7 lysozyme, they should be compatible in nature. To ensure that these two are compatible in nature, you just have to ensure that they do not have the same origin of replication. So that's why here it is written that expression vector cannot have P15A origin of replication, which is actually present in the plasmid P lyses, which expresses T7 lysozyme. Okay. So again means these are the various bacterial strains which have been engineered for various purposes. So here is another one. Here P lysis is here, and here P lyse E is there. But the final aim is to reduce the expression of T7 RNA polymerase so that toxic proteins can be sufficiently expressed in the bacteria. Then there is see, means I'm saying that this is just a limited set of bacterial strains that I'm showing you here, but there are many means hundreds of bacterial strains with different mutations or different forms of mutations for the same genes, but with the ultimate aim of overexpressing recombinant proteins at very high levels. So in this strain, what they have done, it lacks the functional protein called RNase E, and they have found that when they have mutated RNase E, it results in longer transcript half-life. So longer transcript half-life means more amount of transcript at a given point of time, and thus higher amount of proteins. Then there is another strain in which what they have done, they have made it mutated or deleted in RECA protein. So what does RECA protein facilitate is recombination. So it was seen that in many inserts, especially if they have repetitive sequences, if you introduce into bacteria, which is RECA positive, it led to some random recombinations taking place within the vector, within the plasmid, this foreign DNA or the vector, etc. So to make it more stable in nature and ensuring that whatever you have cloned stays in same way, they have made it RECA deficient. Then to ensure, see, wherever you are cloning something, you want for final protein production, you also want that your protein should be folded properly. It should be like very efficient in whatever protein that is being made. It should fold properly, and so that once the folding is proper, then only it will be in functional state. So here what they have done, this particular strain, it contains highly active thioredoxin reductase and glutathione reductase, which facilitate proper folding. So maybe not shown here, but in many bacterial strains, they have also introduced these post-translational modification enzymes. So acetylation, so in one strain, I will be forwarding you one very good paper on these engineering bacterial strains. So in there, specific examples have been given, but in one case, they have actually introduced N-acetyl transferase, this particular gene in bacteria, and they were seeing that the eukaryotic proteins which were expressed from that bacterial strain, they had acetylation, had the desired residue. So similarly, for another modification, they have introduced this whole locus from another bacteria. It was introduced into E. coli, and that particular post-translational modification, it was very well done in the proteins which were expressed. So keeping in mind that bacteria can be an extremely good host, comparatively cheap to grow, and people won't directly want to go to mammalian cell culture based systems, because they are very, very expensive to maintain, difficult to maintain. Their growing time is around 24 hours, etc. People are still trying to engineer the bacteria in such a way that eukaryotic proteins can also be expressed in the way they are actually expressed in mammalian cells. So even those changes have been introduced into certain bacterial strains, and still you will keep finding new and new papers in which new methods are being introduced. So look at this particular strain. See what all scientists are doing to ensure that the protein is expressed properly. Here what they have done, you must be knowing about codon bias, that certain strains or certain organisms have a certain bias towards certain codon, and they have tRNAs which recognize those codons which are prevalent in that particular strain. I'll revisit codon bias when I'll be telling you about expression vectors, but for your information, it is a general fact that different organisms have different bias towards certain codons which give rise to a particular amino acid. So the organism which has a particular codon bias, they might be deficient in the tRNAs which recognize the other codons for a particular amino acid. So here what they have done, they have in one of the bacterial strain, they have added 7 additional tRNAs for rare codons which are not normally found in E. coli. So by this method, even if you express a protein from foreign source into E. coli, it can be efficiently translated because it is not limited by the codon bias. Then another strain has been developed in which they have introduced a method of T7 RNA polymerase based expression only, but it is induced previous was induced by IPTG. So in this it is induced by rhamnose, and their observation was that when you use this system, then inclusion bodies, or let's say protein aggregates, they were much less as compared to in other systems. So they keep trying different methodology to develop bacterial strains which are very good. So this introduction was just to understand and probably admire the fact that how different molecular biologists and working in company or basic labs, they are trying to engineer the various strains for their final purpose, and how different the methodologies can be. So in certain cases, they are doing just random mutation of, let's say a lab has this particular bacteria or any of these bacteria that they are very comfortable using as the host strain. So what they do is they do a random mutagenization by UV rays or some chemical carcinogen, whatever method they are doing. They are trying to do random screening of these strains, because many times we don't even know that what is the limitation of the final protein production. It could be at various levels. Maybe we are not aware of the gene that we need to engineer to have the final protein production at optimum levels. So in those cases, what you do is you just go for random mutagenesis and you screen the various bacterial strains for the strain which gives the best protein output. So even those methods are being used. Or in some cases, let's say you're working on one particular bacteria, you want to use it as a bacterial strain, and there is some other basic study going in another bacteria in which they find a gene which is very important for, let's say, transcript stability or protein stability or for certain modification. So with that knowledge, you try to use that in the bacterial strain or even yeast strain that you're working on, and then try to engineer that gene in your strain of interest, and then see whether it is making your final output good. So when we are talking in terms of industry, at least, see, for your basic production, even if you get little less protein output, or even that would be maybe sufficient for your daily molecular biology experiments. But from industrial point of view, even if you increase it, even 1.5 fold protein production by any of these changes, it is a huge profit to the industry. So obviously 1.5 fold, I've taken it from the lower side, but you aim to get it like 10 to 20 or 100 fold higher expression than the original bacterial strain. So I guess that was a comprehensive introduction to how do you engineer bacterial strains, and I've already told you that I'll be forwarding you one research paper for the same. And now coming to introduction to the various methods, or let's say various vectors which are used for bacterial host strains, and those are plasmids and those are bacteriophages. So first, let me get started with plasmids. So I'll just give a brief introduction of what plasmids are and what are the important properties of plasmids, which are very important in terms of using them as vectors for cloning. So yeah, so first, general introduction. These are extra chromosomal entities. They are generally circular in nature, though there are some rare cases in which you will also find plasmids which are linear. So the examples are given here, but in general, we find circular plasmids, and most of the vectors that are present in the market, they are circular in nature. These plasmid vectors. Then there are various ways by which plasmids can be classified. I'll just rush through the basic introduction to plasmids, mainly due to the fact because you have been already exposed to what plasmids are and their properties in microbiology class. So one point is that, of course, on the basis of whether these plasmids can be transferred from one bacteria to another, you see whether they are, they can be classified as conjugative or non-conjugative. So conjugative ones, they generally get transferred by a pilus. And one important point when we are talking in terms of plasmids to be used as vectors, you generally want that they should be non-conjugative in nature. And why do you want them to be non-conjugative is due to the fact because you do not want the plasmids which have been the recombinant plasmids in which you have cloned your gene of interest into bacteria to be spread into the environment. You do not want that. You want it to be highly contained in nature because you do not know that if you spread that particular gene of interest which you have maybe made as recombinant, or even a gene sequence which is not normally found in the environment, you do not want it to be spread into the environment. So generally in that context, plasmids you prefer as non-conjugative in nature, or the plasmids which have very maybe narrow host range. yeah, I'll come to that about the host range. In some cases, you may want a broad host range also, but generally they should be non-conjugative in nature. Then plasmids can also be classified on basis of functions, and in that scenario, I'm sure you know all of these terms which are mentioned here. They are fertility plasmids capable of conjugation. They are plasmids which provide resistance to certain antibiotics, to certain bacterial strains. They are plasmids which code for colicins that can kill other bacteria. They are plasmids which can degrade certain substances, and then there are virulence plasmids, etc. So these are just certain facts about plasmids. One very important point with respect to plasmids to be used as vectors is their size and copy number. So generally, you prefer a big plasmid vector which is very small in size. So why am I mentioning that is because plasmids which are naturally occurring, they range from, let's say, 0.7 kb to more than 200 kb. Okay, so generally you will prefer a plasmid vector which is very small in size. Why would you prefer a plasmid which is small in size is due to the fact you want that your the cloning capacity, that is the size of the foreign DNA that will be incorporated into the plasmid, it can be as big. I mean, because the plasmid that can be transformed easily into bacteria can be around 10 kb. So if you have, let's say, a plasmid which is 8 kb, then its cloning capacity is only 2 kb. So that's why you prefer plasmids which are small in size and have all the other properties which are required for it to be vector, and can accommodate much larger inserts. So one point is that with respect to size, and another point is the copy number. So I have told you, so these are naturally occurring plasmids. Many of these plasmids are present in very low copy, even 1 to 2 copies per bacterial cell. And there are other plasmids which are present in very high copies. So when we talk in terms of more production of a protein, then you prefer the plasmids which have high copy number. So because more of the copies are there, more transcript is being made, and ultimately more protein is being made. So in all scenarios, do you prefer a very high copy number plasmid? Maybe not. In some scenario, when your protein is a little toxic or highly toxic, then probably you would like to clone it in comparatively low copy plasmid vector. Okay, so these points will be important for your individual need of cloning a particular DNA segment. And what is your ultimate aim? Is your ultimate aim to get it being just maintained as in a plasmid vector for your later use, or do you want to finally get more of recombinant protein? So in that context, maybe you will require higher copy number. Now, what are the various mechanisms by which copy number of a plasmid is regulated is one point that you should be aware of, because unless you are aware of these points, you might not be able to understand that what changes have been have been done in plasmid vectors to make them as good vectors. So before going to the mechanisms, I want to know whether you have been taught this either in your microbiology class or in your molecular biology class. Are you aware of these two mechanisms or not? I can't hear anybody.
No, ma'am.
So none of you? Because then only I'll go through it. If you have already gone through it, I'll not even rush through it. Okay. So there are two major methods which have been, known or studied with respect to which methods bacteria uses to regulate the number of plasmids which are present in that bacteria. So the most commonly used is antisense RNA, found in many bacteria, but then in some there is something called as iterons. Even that is the way by which copy number has been regulated in plasmids. And if you read literature, maybe you'll find many more methods also, but these are the two common methods, and that's why I'll be just telling you that what is this method. Okay, so first coming to antisense RNA mediated method of control of copy number of plasmids. Okay, I think this is the one which is present in coli-1 plasmids. So let's see this particular figure and focus on RNA-pol2, RNA2 transcript. Okay, so this RNA2 is the transcript which is important for initiation of replication of the plasmid. So if you ever see an arrow like this, it means that the promoter of this particular transcript is here, upstream. It means that the promoter for this particular gene is here. Okay, so you should always be aware of the arrow thing. Here the promoter is here. Okay, so anyway, the promoter is here, and let me just tell you first about RNA2 transcript. So when RNA2 is made by a promoter which is present here, it assumes some secondary structure here, and the 3' end of this RNA2 transcript, it reaches the ORI site, which is present few bases away from this RNA2 transcript. So once this transcript reaches the ORI site, you know that in ORI site it opens up. So this is a bubble, replication bubble, which is present here. So the 3' end of this transcript, it is acted upon by RNase H enzyme. If you remember my previous class, I've told you that RNase H enzyme is the enzyme which cleaves the RNA in the RNA and DNA hybrid. So it cleaves the 3' end, and this free 3' OH group that is now generated is recognized by DNA polymerase 1, which is recruited here, and then it leads to means this acts as a RNA primer, and this is the free 3' OH group, and then it starts replicating, means it starts adding the nucleotides with this as the template. So this whole process leads to replication of the plasmid. Now consider another scenario in which we have lots of plasmid which has been now generated within a bacteria. So RNA1 transcript is the transcript which is generally present in very low level, but when we have huge amount of plasmids which is accumulated in the bacteria, then what we have is comparatively much more amount of RNA1 being produced. Now if you can see that the 5' end of this green RNA2 transcript is complementary to RNA1. So it is made, so it is an antisense, its promoter is here, and the transcript is made like this. Okay, so this region is complementary. So when we have high level of RNA1, then we have these two transcripts, the RNA1 and RNA2, which are made in the cell. They form a very weak kissing complex. It's called a kissing complex. I don't know who named it. Anyway, so they too make a complex which is comparatively having a weak interaction. But then there is one protein called ROP which goes and binds to this kissing complex and makes it much more stable in nature. Once it stabilizes this particular complex, then it leaves the complex, and what happens, we have a RNA1, RNA2 duplex being made, because of which we don't have this priming action. RNA2 is not available for the priming action, and no replication takes place. So now you can understand that when a plasmid reaches a high copy number and it should not be increased beyond that copy number, then RNA1, RNA2 interaction, which is stabilized by ROP, that gets formed, and we don't have the final priming reaction, and plasmid replication stops. It means the copy number is regulated. Now let's say that I find a plasmid vector which I am very interested to use, but it has low copy number. So by which way I can increase its copy number? Can you think of some way by which I can increase its copy number, considering that, let's say, that plasmid is dependent on this method of copy number control? Okay, so if you are given this task of controlling copy number for that particular plasmid, what changes will you make in that plasmid so that it has much higher copy number?
We can inhibit the formation of RNA1.
You need to be louder.
We can inhibit the formation of RNA1.
Okay, so how can you inhibit the expression of RNA1?
Then we can have specific binding proteins so that it cannot be expressed.
Okay, so you go for specific binding proteins for RNA1 so that it is unable to bind to RNA2.
Yes, ma'am.
Okay. And yes, that can be one theoretical possibility, though it is generally not used. They have either made a deletion or mutation in RNA1 so that it is unable to bind to RNA2. And yes, there can be many other ways. Like he said, you can think of the ways. Let's say you are in a any lab or company and you are given various tasks कि आप इस final आपको एक plasmid दिया जाएगा and you will be asked to increase its copy number. So you can use, maybe you can use a specific chemical which binds to RNA1 and doesn't let it bind to RNA2, because you know that this is the point that you want to inhibit. Any other method that you can think of? So in many vectors, what they have done, they have made ROP mutants or deletions. If ROP is not there, then this complex is not stabilized, and ultimately RNA2 is available for priming function. So, and there is no control of copy number in that case. So in many vectors, you will find either RNA1 or ROP mutations or deletions. Okay. Similarly, there is another method which is iteron mediated. So what are iterons? They are nothing but these sequences. These are repeat regions which are present in the origin of replication. So just let's look at this point. So there is this ori region, and these are the three iterons. So there are two ways by which it was found that the copy number was controlled here. So let's consider that what is the normal way of replication in this particular plasmid. It is done with the help of repe protein, which when a protein is formed, it goes and binds to origin of replication and brings about the replication of this particular plasmid. Now, let's say we have very high amount of repe being used, then one method by which it can inhibit further replication of the plasmid is by auto inhibition. So what it does is, this protein, once it is present in very high amount, it goes and binds to its own promoter and inhibit further replication of, sorry, transcription of repe. So less repe will be produced. So this is one method. And in another method, what happens of inhibition, that once we have high amount of repe that is being produced, it goes and binds to these iteron regions which are present in the ori, and this par region means partition locus which is present here. Once this repe binds to these particular regions, so it will bind, consider that there are multiple molecules of plasmid in your this whole bacteria. So, and they are bound by repe proteins. So what will happen, because repe, it forms a dimer. So what it does it, that let's say this is one plasmid and this is another plasmid, both of which are bound by repe proteins, because repe protein, let's say, it's present in huge quantity because we have high plasmid copy number. So then it will handcuff the two plasmids, and because the two plasmids are now handcuffed together or locked together, there will be no further replication which will take place. So this is another method of control of copy number in other plasmids. So depending on which host strain you are using, it's not always E. coli. In many cases, you work on various different bacterial strains, and you want to introduce the bacteria which is maybe native to that bacterial strain, and make a recombinant plasmid in that. So you have to first see how its copy number is regulated. Once you know how its copy number is regulated, then you can of course make changes in the vector to ensure that higher copy number is there. Another way to control the copy number of a particular plasmid was by treatment of chloramphenicol. And in this case, what they have done is that let's say there was one plasmid which is present in around 20 copy number. You want to drastically increase the copy number to, let's say, around 1000 or 500 copies. So what they have done is they have given a treatment with chloramphenicol. What chloramphenicol does is it inhibits the translation. It inhibits the translation. If the translation or protein expression is inhibited, then it was seen that for some time the plasmid, it continues to replicate. It is unaffected by no fresh protein synthesis, because the proteins which are required for its replication is quite stable in nature and they stay for some time. But it was seen that the bacterial chromosome replication was inhibited strongly. So now the whole energy goes in replication of the plasmid, and bacterial chromosome is not replicated. As a result, what they found that after treatment with chloramphenicol, a huge amount of plasmids were present after the treatment. So what they generally do is they grow the bacterial host strain for a particular density. Once it reaches a particular density, they give a treatment with chloramphenicol for some time, which inhibits the protein synthesis, because of which this bacterial DNA does not get replicated, while this gets replicated to huge amount, and we get means a huge difference in the copy number of a particular plasmid that you are interested in. So different methods have been used to increase the copy number. Maybe not all plasmids are sensitive to this. We don't know. So for individual purposes, you'll have to optimize the way by which the copy number of a particular plasmid may be increased. What's the time?
9:55.
Okay, so then I will continue in my next class. We'll meet tomorrow, and you may ask any question if you have. Are you understanding?